Assalamualaikum warahmatullahi wabarakatuh. Hello guys, how are you today? I hope everything is going well. Okay, before starting my video, let me introduce myself. My name is Risza Nuril. I'm from Biology Education, offering A number 30. Well, in this video, I will deliver a journal and it's about this picture. So, do you know this picture, guys? Well, it's right. It's placenta. And the title of our journal in this video is Placenta and Placental Derivatives in Regenerative Therapies. So actually, what is placenta? Okay, in simple definition, placenta is an organ that connects the mother and the baby, so the baby can get nutrient, gas, and other supply from the mother. Placenta is a unique organ. Why? Because it's a temporary organ which ensures mutual coexistence of the organisms between the mother and the fetus. And placenta also has function to ensure the supply, growth, and development of the fetus, as well as removing metabolic products and fighting against internal infection. Another unique of placenta explained in the journal is that researchers found that placenta and placental derivatives has many stem cells that can differentiate into other types of cells. So here, placenta as a stem cells can be medicine as regenerative therapies for kinds of disease. Why? Because it can differentiate into other types of cells. Okay, next. So guys, actually what is stem cells? Well, stem cells are cells that can differentiate into other types of cells and can also divide in self-renewal to produce more of the same type of stem cells. And placenta as a stem cells, it means that placenta has ability to differentiate and become other kinds of cell. That's why placenta can be a regenerative therapy for kinds of disease. So I will show you the picture. It's a placenta, and as a stem cell, it can differentiate into the pancreatic cell, hematopoietic cell, cardio cell, neurons, and also hepatocytes. And now I will explain you the morphology of the placenta. The postpartum placenta has a disc shape from 16 to 20 centimeters in diameter and weighing 500 grams on average. So here I will show you the picture. It's a disc shape. So the shape is like disc. And it has three main cell types: the trophoblast cell, mesenchymal cell, and endothelial cell. The trophoblasts are cells forming the outer layer of the blastocyst, which provide nutrient to the embryo and develop into a large part of the placenta. And the next is the mesenchymal cells. It's a multipotent stromal cells and can differentiate into a variety of cell types. So, because the placenta has these two kinds of cells, that's why placenta can be stem cells, is having many stem cells, and can be a medicine for regenerative therapy. And the next is the epithelial cells. It's the outer cells of the placenta. The placenta for research in this journal is here. There are two kinds, the early placenta and the mature placenta. The early placenta from the first 2 trimester of the pregnancy, while the mature one is from the 3rd trimester, and it has developed the fetal and the maternal parts. The researchers use the mature placenta. So in this journal, it's explained that the researchers use the mature placenta because it has been developed, yeah, for the fetal part and also the maternal part. So here the picture of the part of placenta. The maternal part is the part that attached to the mother, while the fetal part is the part that attached to the fetus. Well, in pregnancy, the function of placenta is for immunological features, endocrine function, nutrition, gas exchange, excretion, produce hormone that support the pregnancy, and the thermal regulation. So guys, here, because the placenta is used for regenerative therapy, it's mean that we have to save placenta for a long time. And here the methods that researchers use for saving placenta. The biobanking methods of placental components. The first is by sterilization. So we sterilize the placenta. We before saving them, the placenta must be sterilized first from the germ, from the bacteria, and others. And the next is it must be saved in low temperature storage. And in the saving of placenta, researchers use this method, cryopreservation. Cryopreservation is the most conventional method for storage of biological objects, and it provides high levels of preservation for a long period of time. So guys, because the placenta is needed for a long period of time, so it must be saved in the low temperature storage and sterilized and saved in this method. It's the method that researchers used. Okay, well, those all my presentation about the journal. The conclusion is that placenta has many stem cells that can create other types of cells from other organs. So placenta can be a medicine for degenerative sickness or disease, and it can be regenerative therapy for kinds of disease. Thank you so much. I hope this will give beneficial for you all, and wassalamualaikum warahmatullahi wabarakatuh.